Dear students, Assalamu alaikum. We were discussing about the structure of the DNA. DNA has a compact double helix structure. In this structure, I told you that 0.34 base pair distance between the two bases, and in a single turn there are the 10 base pair, 10 bases in a single turns are present. So the total distance in a single turn is 3.4 nanometers. Other than the distance, there are two grooves in the DNA. One is the minor groove, and second one is the major groove. And these major and minor grooves are formed because of the two glycosidic bonds are not diametrically opposite each other. Each base pair has a larger side that defines the major groove and has a smaller side that defines the minor groove of the structure. The groups are lined by the potentially hydrogen bond donor and hydrogen bond acceptors. The floor of the major groove is filled with the base pair nitrogen and oxygen, which are pointed or project inward from the sugar phosphate backbone toward the center of the DNA. And the floor of the minor groove is filled with the nitrogen and the oxygen atoms of the base pair that project outward from the sugar phosphate backbone toward the outer edge of the DNA. There are three major forms of the DNA: B, A, and the Z form. In the B form, the helix is more compact. The helix has the more compact structure. In the B form, the helix is almost somewhat relaxed structure. It contains the 3.34 distance between the two bases and 10 bases in a single turn. So the total distance between so the total distance of a single turn is about 3.4 nanometers. While A form is more compact, 11 bases in a single turn and 2.3 nanometer total distance. And the zDNA is actually the compact DNA and 12 bases in a single turn. It is more compact DNA. It makes the zigzag form of the DNA, so that's why it's called the zDNA. So it may play a role in the regulation of the DNA. Although the multitude of the hydrogen and hydrophobic bonds between the polynucleotide strands provide stability to the DNA, the double helix is somewhat flexible about its long axis because unlike the alpha structure in the alpha helix in the protein, there are no hydrogen bonding between the successive residues. This property allows the DNA to bend, for example, when compressed. Here against the chart, so helix direction. Uh, Helix form A, B, and D, helix direction. A is the right-handed, D is the right-handed, and Z is mostly the left-handed DNA. So the base pairs per turn in the A are the 11, 12 in the Z, and the 10 bases in the Z form of the DNA. And these are the scientists Watson, James Watson, Francis Crick, and Rosalind Franklin. Uh, who are the major players. Watson implies Wilkins, Maurice Wilkins, Francis Crick. So these are the major players to determine the structure of the DNA. Tertiary structure of the DNA, like the other molecules, DNA has also the tertiary structure. The second structure, the secondary structure of the DNA has described as a double helix. Is it possible for this already highly organized structure to twist into more complex arrangements, as we observed in the three-dimensional structure of the protein? Genetic studies and the electron microscopy on the native and intact DNA have revealed two distinct forms, linear and the circular. So this is very important to know that the DNA has the tertiary structure to be more compact and compact to reside in the in the cell. The chromosomal DNA of many microorganisms, however, is a closed circular, a result of covalent joining of the two ends of a linear double helix. Closed circular duplex DNA has a unique structural feature. It is found twisted into new conformation supercoiled DNA, and it makes the three-dimensional structure. This is the say for example, this is the double stranded native molecule. Uh, when we heat this molecule, the double helix will be broken down into the into the two strands. If you create a nick, these two strands can be separated from one another. And this is the electron micrograph of the relaxed and the supercoiled DNA. Here you can see that the DNA is present in the supercoiled form, so the tertiary structure of the DNA is possible. The supercoiled form is less stable than the relaxed form. However, there is a sufficient evidence to conclude that the supercoiled DNA exists in vivo. And the relaxing feature of the DNA is very important for regarding the expression of the DNA. Regarding if it remain in the complex form, it remains in the compact form, it cannot transcribe. So relaxed feature is important for transcription of the DNA. The most convincing evidence is the isolation and characterization of the protein that catalyzes the interconversion of the relaxed and the supercoiled DNA. These proteins are called topoisomerases because they catalyze the change in the topology of the DNA. This is very important. And these are the very important enzymes which make the DNA relax and decompact time to time. So this is the relaxed DNA, and second B diagram is for the supercoiled DNA. The cause of supercoiling is not completely known, but it may be the presence of more than standard number of base pair unit length of a DNA helix, forcing tension in the molecule. Supercoiling may have a biological significance in that DNA molecules become more compact, and hence it is more easily stored in the cells. The next molecule is the RNA. RNA molecule. What will be the structural features of the RNA? Now we need to discuss about the RNA. The next topic will be the RNA structural element, but before going towards the RNA, we will have a little bit discussion about the chromosomal structure. First, we start from the bacterial chromosome. The bacterial chromosome is found in a region called the nuclei. The nuclei is not a membrane bounded, so the DNA is in direct contact with the cytoplasm. Um, Bacteria may have 1 to 4 identical copies of the same chromosomes. The number depends on the species and growth conditions. So, bacteria do not contain a well-defined nucleus in their cells. This is the feature of the prokaryotic cells. Bacterial chromosome DNA is usually a circular molecule that is a few million nucleotides in length. Say, for example, in the E. coli, for nearly approximately about 4.6 million base pairs are present in the DNA of the E. coli. In the influenza, around 1.8 million base pairs are present. A typical bacterial chromosome contains a few thousand different genes. These genes may contain the structural gene sequences, the non-transcribable DNA. The structural DNA sequences are the sequences which can code the proteins. So, protein coding genes are called as structural genes, and the non-transcribable DNA, which cannot be described into any protein, which cannot be translated or transcribed into the protein. This is, say, for example, the chromosome of the DNA. It starts here. This region is from this region. This is the origin of application region of the bacterial genome, and these are the non-transcribable regions, and this one is the transcribable region of the DNA. Most but not all the bacterial species contain the circular chromosomal DNA. A typical chromosome is a few million base pair in length. Most bacterial species contain a single type of chromosome, but it may be present in multiple copies. Several thousand different genes are interspersed throughout the chromosome. One origin of replication is present in the DNA. This region is responsible to replicate, to initiate the replication in the replication of the DNA. Short repetitive sequences may be interspersed throughout the chromosomes. These are the short interspersed sequences. These play a role in DNA folding, DNA replication, and gene expression. fit within the bacterial cells, the chromosome DNA must be compacted about 100 folds. This involves the formation of the loop domain. Here, this is the circular DNA of the bacteria, and this is the loop chromosomal DNA with associated proteins. The loop structure compact the chromosomes about 10 folds. The number of loops varies according to the size of the bacterial chromosomes and the species. E. coli contain 50 to 100 with 40,000 to 80,000 base pairs of the DNA in each. DNA supercoiling is the second important way to compact the bacterial chromosome. After the supercoiling, after the making the looped chromosome, the supercoiled, coiled and looped DNA is formed. The supercoiling within the loops create a more compact DNA. The control of the supercoiling in the bacteria is accomplished by the main enzyme DNA guidase, also termed as the DNA topoisomerase 2. Because of this enzyme introduces negative supercoils using energy from the ATP. It can also relax the positive supercoil when they occur. DNA, and the second enzyme is the DNA isotopoisomerase 1, which relaxes the negative supercoiling. The compact action of these two enzymes governs the overall supercoiling of bacterial DNA. You can see this one is the relaxed form of the DNA, and this one is the negatively supercoiled covalently closed DNA. Information about the structure of the DNA has come from the studies of the prokaryotes because they are less complex, both genetically and biochemically, than the eukaryotes. Prokaryotes are monoploids. They have only one set of the genes. In most virus and the prokaryotes, a single set of gene is stored in a single chromosome, which is in turn contains a single molecule of the nucleic acid. The smallest known RNA virus have only three genes, and the complete nucleotide sequence of the genome of many viruses are known. For example, the single RNA molecule in the genome of the bacteriophage consists of 3569 nucleotides and contains the four genes. Complete nucleotide sequences are known in several cases. For example, the genome of bacteriophage is X171 is a single DNA molecule 5368 nucleotide in the length that contains 11 genes. The largest virus, like the T2, and the animal pox virus contain about 150 genes. Bacteria like the E. coli has 2500 to 3500 genes. In the past, prokaryotic chromosomes were often characterized as the naked molecule of the DNA. In contrast to the eukaryotic chromosome with their associated proteins and the complex morphology, this misconception results in a part because most of the published picture of the prokaryotic chromosomes were electron micrographs of isolated DNA molecules, not metabolically active or functional chromosomes, and most of the published photographs of the eukaryotic chromosomes were highly condensed meiotic and mitotic chromosomes, again metabolically inactive chromosomal states. Functional prokaryotic chromosomes are nucleoids, um, nucleoids rather than the nuclei because they are not enclosed in the nuclear membrane. Again, telling you, in past, the prokaryotic chromosomes were often characterized as the naked chromosomes, in the past, the DNA was the prokaryotic DNA was known as the naked DNA. It was because the electron micrographs of the bacterial and the eukaryotic chromosomes was in the compact stages. So that's why this was the misconception that the bacterial DNA is naked and the eukaryotic DNA is not naked. The counter length of the circular DNA molecule of the bacterium E. coli is about the 1500 micrometer because E. coli cells has a diameter of only 1 to 2 micrometer. So, because bacterial cells is almost about 2 micrometer and the length of the DNA is about 1500 micrometer. So, this is not possible for the cell to reside the DNA into them without supercoiling of the molecule. When E. coli chromosomes are isolated by gentle procedure, uh, in the absence of the ionic detergents commonly used to lyse cells, and are kept in the presence of the high concentration of cations such as the polyamines or 1 molar salt to neutralize the negatively charged phosphate groups of the DNA. The chromosomes remain in a highly condensed state comparable in size to the nucleide in vivo. This structure, called the folded genome, is the functional state of the of bacterial chromosomes. Though smaller, the functional intracellular chromosomes of the bacterial virus are very similar to the folded genome of the bacteria. Within the folded genome, the larger DNA molecule in an E. coli chromosome is organized into 50 to 100 domains or loops. Um, Within the folded genome, larger DNA molecule in the E. coli chromosome is organized into 50 to 100 loops, each of which is independently negatively supercoiled. RNA and the proteins are both components of the folded genome. Because each domain of the chromosome is independently supercoiled, the introduction of strand nick in the DNA by treatment of the chromosome with the DNase that cleaves the DNA at internal site will relax the DNA only in the naked domains. Destruction of the DNA connectors by the RNases will unfold the folded genome partially by eliminating the organization of the DNA molecule into the 50 to 100 loops. However, RNase treatment will not affect the supercoiling of the domain of the chromosome. So these are the features of the folded genome. First, the circular DNA is organized into 40, 50 to 100 loops, and further, each loop contains its supercoiling structures. RNA and proteins are both components of the folded genome. These are also attached with them, which can partially relaxed by treatment with either DNases or RNases. Because DNA contains the loops. When the nick is generated, only the nick part will be relaxed. Here you can see this is a circular DNA, and it contains 40 to 50 loops or 50 to 100 loops whatever. And after making the loop, the size of the chromosome is reduces from 350 micrometer to the 30 micrometer. Further, the supercoiling reduces its size to the 2 micrometer, and it reduces the size up to the 2 micrometer. Each loop is independently supercoiled. When we treat it with the, say for example, DN, when we cleave it to make it relaxed, say for example, this is the nicked DNA. And then this region is actually the partially uncoiled chromosomes. This one is the partially uncoiled chromosome. So, for example, here are some RNA molecules. When RNA is cleaved, uh, so it make it relax. Partial RNA digestion, partial unfolding of the chromosome will be created. And if we treat it with the DNase digestion, then we treat it with the partial DNase digestion, it will partially uncoil the chromosomes. There are two differences. Treating with the RNA, partial unfolding of the chromosome will take place, while treating with the DNase, partial uncoiling of the DNA will take place. This is the structure. This is the overall structure of the prokaryotic chromosomes. The next topic will be the organization of the eukaryotic chromosomes. Tomorrow we will discuss about the eukaryotic chromosomes. Good luck.